Hi, welcome to the channel. In this video, I'm going to teach you the bacteria artificial chromosomes and yeast artificial chromosomes. So, we can discuss this topic. And what are bacteria artificial chromosomes? These are the DNA construct based on functional fertility plasmid or F plasmid. Say they are formed from the F plasmid. These are generally a DNA sequence or the DNA structure which is formed from the functional fertility plasmid. These are the reproductive plasmid in bacteria. These are BT artificial chromosomes used for transforming if they can insert into a bacteria. They provide certain characteristics to the bacteria there, so that the bacteria gets transformed, and it has been cloning bacteria. Whenever the bacteria cell divide, it also replicate and increase copies and transfer it to the another bacteria. Part dividing bacteria. They're capable of carrying approximately up to 300 kilobase pair of inserted DNA sequence. It means they carry the gene of interest, which is of maximum size of 300 kilobase pair of. Now, how is its structure? This is your BAC 108L. It has certain size or inside it. So, common gene components in bacterial artificial chromosome. So, replication E-RAPI for plasmid replication, regulation of copy number. RAPI is a site which regulate the copy number, how many plasmid number will form inside a bacterial cell for plasmid replication. And PAR A and PAR B are the partitioning of plasmid. They help on whenever the cell is dividing. It ensures that the bacterial artificial chromosomes number also get transported into daughter cells. Now we have selectable markers. These are the selectable markers which help us in finding out whether the cell gets transformed. It means it gets plasmid with gene of interest, or it does not get the plasmid with gene of interest. So it has these selectable marker like antibiotic resistance and lac jet as at a cloning site for blue-white selection, which helps us in determining whether the cell is transformed or not with a recombinant DNA. Now we have T7 and sp6 site, which is phase promoter for transcription of inserted genes. If when the gene of interest gets inserted, then this gene of interest is expressing itself, like it is forming a RNA, messenger RNA. So that's the transcription is happening. This is these are regulated by the promoter, that is T7 and sp6. Now we have 4IS. These are the origin of replication. This is the place through which the replication of this plasmid occur. Okay, so you can see these all the sites here, like origin of replication is. Now replication E, which have been regulating the copy number, like how many copies of this plasmid or this bacterial artificial chromosomes will form. And now we have pair PAR A and PAR B, which help and ensure like the partitioning when the cell is dividing. It ensures the install maintenance of BAC for partitioning of plasmid DNA. अच्छा now okay, now we have molecular cloning in bacterial artificial chromosome. This is showing the complete overview how this bacteria artificial chromosomes work. If you see this, here we have the foreign DNA from the top of the diagram. Then we have plasmid, which has a restriction site. Restriction site is a site through which the restriction enzyme can cut the plasmid. Now we have two important selectable markers, lag gene, and then we have ampicillin resistance gene. Now here the restriction gene cut the restriction site at particular sequence like GG sequence you can see here. Now when they cut, so it form a stick ends, and in those stick ends we are going to place our foreign DNA, which is our gene of interest, which we need that it replicate and it produce its product or through transcription or through translation both. So now we have added these gene of interest in these um, plasmid. Now the plasmid contain our gene of interest. Now here you can see the bacteria. Up now when we are placing bacteria and we are trying to incubate the bacteria, and we're trying to insert this plasmid of having a gene of interest inside a bacteria. So there are so many circumstances can be happening. You can see here bacteria may take a plasmid with or without the insert, or may not take up the plasmid at all. So there's when you're trying to put plasmid inside a bacteria, there are so many chances. So the first probability, like it will not take plasmid, as you can see in this picture. Okay, in the second picture from left side, this does not contain any plasmid. It means this bacterial cell does not able to insert the doesn't able to uh, get on the plasmid. And you can see in this second one, bacterial genome is missing the lecture gene. Here you can see the bacterial genome is missing the lecture gene. It doesn't contain lecture gene, so when we insert the plasmid, then it contains the lecture gene. In the third one, you can see it has may take up plasmid with or without the insert. And what happened that in the in this case you can see here, like plasmid is inserted but the plasmid does not contain our gene of interest. And the last one you can see the cell has the plasmid, and the best thing that the plasmid is transformed. It means it contains our gene of interest. So this is the best cell for us, because now this bacterial cell will going to produce our gene of interest protein. Now you can see, now those bacteria cell which contain like Z gene plasmid, then these are going to be so different uh, colonies. So the transform one will show white colonies have plasmid with the foreign insert. So those which cells which are showing white colonies, they have the plasmid which have the foreign gene insert, because we have placed this foreign gene of interest inside a lecture gene, jet gene. So lecture gene is not able to express itself when there is a gal, then it is not it's not able to produce beta galactosides. So if the beta galactoside is not producing in this cell, then it's not going to digest the beta x gal, so blue color is not going to produce. But in those plasmid which do not have uh, our plasmid having gene of interest, the plasmid gets enter, but inside this plasmid we do not have our gene of interest. So blue cornel is formed because it is normal, because lecture gene is not insertionally inactive in these colonies, so it will produce beta galactosides, and xcal will get um, digested, and eventually it produce blue color, that is a iodide color. Okay. Application of BAC. Sorry, I haven't taken the screenshot here. And today. And so bacterial artificial chromosomes are generally used in modeling modeling genetic diseases in order to study the effects in experimental transgenic mice. So what we did with that bacterial artificial chromosomes. So these are very helpful in studying the facts in experiment when we are doing the experiment on the transgenic mice. We can study various genetic diseases with the help of BAC because it is very useful in modeling genetic disease. Um, BAC has been used in neurological diseases such as Alzheimer's disease. In the case of Down syndrome, we can study these diseases with the help of BAC. The genome of several large DNA viruses and RNAs have been cloned in BAC. We can have study genomic library. So these are very helpful in creating the genomic library and it is very helpful in human genome project. They construct various genome of several large DNA viruses and have been cloned as pictures across chromosomes. Genome clone code. These constructs are referred to as infectious clones. Let me repeat it. The genome of several large DNA viruses and RNAs have been cloned as BACs and clone किया इसको infectious clones. Uh, I hope you are able to understand. This is the lecture on the BAC. I have also watched so many videos, which is very helpful for me to get a good idea of this topic. If you really wanted to watch, you can also watch because it will clarify so many doubts of yours. Now I'm done with this BACs.